Hey there, sweetheart. I was so excited to get your call and learn that you've been sharing my letters with your friends in science class. Maybe all of you can gang up on the professor and show him the error of his ways. Well, I'm just kidding about that. But tell your friends that if they have questions, I'll be happy to try to answer them too. Well, I hope you're agreeing by now that the evidence is very strongly pointing toward intelligent design as the cause and force behind the universe and all of creation. If you or your friends still have any doubt, this letter should quash it for good and make you a firm believer in intelligent design. In this letter, we'll talk about DNA. Remember that when Charles Darwin wrote The Origin of the Species, he thought a cell was nothing more than a droplet of jelly. He called it protoplasm, and he had no idea how complex a living cell actually was. And while he could observe that offspring had the same traits and characteristics as their parents, he had no idea what reproductive mechanism caused that. Research progressed as technology produced better and better microscopes. Through the late 19th and early 20th century, scientists came to grasp the enormous complexity of the living cell. And they realized that each cell contained the instructions for reproduction. But they still had no idea what it looked like or where to find it. By mid-century, many teams of scientists were aggressively searching for those answers, realizing that this discovery would forever immortalize them in the annals of science. In 1953, James Watson and Francis Crick discovered the double helix structure of the DNA molecule. And since the 1960s, every scientist and scientific publication has recognized that DNA is this: coded information. And more importantly, everyone now understands and accepts that information is what runs the show. For every living thing, from single-cell bacteria to every fish or fowl, plant or animal, massive amounts of information compressed into sophisticated code governs all aspects of life. Well, how much information, you might ask? Your personal DNA sequence would fill 200 1,000-page phone books. You know, I'm wondering if you've ever even seen a White Pages phone book. Maybe not. So let me say this another way. If you could type 60 words a minute, 8 hours a day, it would take you 50 years to type out your DNA sequence. And yes, that is a lot of information. And every cell in your body contains the full set. You have 40 trillion cells in your body. Well, by now you should be asking yourself, if every living thing runs on this unique set of instructions written in DNA code, where did all that information come from? Remember, a single cell bacteria needs 300,000 to 500,000 base pairs or bits of code to form its DNA in order to be alive and reproduce. Who wrote and downloaded that software? Well, the materialists have only one answer. All a big accident, blind luck, trial and error. But looking at the variety, the complexity, and the symmetry of all kinds of life, does that really seem like a reasonable, plausible answer? Okay, let's jump into the weeds just a bit so you get a small sample of how complex and complicated this information processing system is.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Now we have another big question to ask. Where did the technology come from to read and process all of that information? Was that just another accident of nature? I want to change direction for a few different comments. You'll remember how we saw earlier that the pursuit of scientific knowledge is a human activity and prone to mistakes. It's fair to say, I believe, that while scientists are on the path to full knowledge, wherever they are today on that path, their knowledge is still incomplete and could even be wrong. I make that point as an introduction to a couple of short videos I want to show you next. You met Richard Dawkins earlier. He's one of the leading atheist voices all over the world and has stated unequivocally that Darwinian evolution is as much of a fact as the Earth rotating around the Sun. In this first short clip, Dawkins is making fun of God and creationists, and then makes a statement of fact that pseudogenes are nothing more than junk DNA left over from a time in the far past when they actually did something useful. In other words, evolution has moved us past the need for these codes, but they still sit in the system as useless junk. Listen carefully for that as you watch.
Forward TV. The world is thinking.
Do you think there could be one sentence that could convince, let's say, a creationist to seriously doubt their theory? Ideally, if you could convince a believer in God to really doubt their belief, but that's too hard.
Not sure about a sentence. I think perhaps the single most convincing fact, the observation that you could point to, would be the pattern of resemblances that you see when you compare the genes using modern DNA techniques, actually looking at the letter-to-letter correspondences between genes. Compare the genes of any pair of animals you like, pair of animals, pair of plants, and then plot out the resemblances, and they fall in a perfect hierarchy, a perfect family tree. And the only alternative to it being a family tree is that the intelligent designer deliberately set out to deceive us in the most underhand and devious manner. Moreover, the same thing works with every gene you do separately, and even pseudogenes that don't do anything but are vestigial relics of genes that once did something.
That clip was recorded in 2009. This next one was recorded less than 5 years later. And in this one, Dawkins admits that pseudogenes are not junk at all, but actually perform necessary functions.
Put your trust in the scientific method. Put your faith in the scientific method. I have enough faith in the scientific method.
You know, when in 2000 the genome was decoded, it was discovered that only 2% of the genome actually codes for proteins, and as you know, the other 98% was dismissed as junk DNA. Last week, 6 days ago, the result of 1,600 experiments by 450 scientists in 32 different institutions discovered that actually that 98% that people thought was junk isn't junk at all. It's absolutely essential for the maintenance of life.
Fascinating fact that the chief rabbi has, the evidence he's given, that whereas we thought that only a minority of the genome was doing something, namely that minority which actually codes for protein, and now we find that actually the majority of it's doing something. What it's doing is calling into action the protein coding genes. So you can think of the protein coding genes as being the sort of toolbox of subroutines, which is pretty much common to all mammals. I mean, all mice and men have the same number, roughly speaking, of protein coding genes, and that's always been a bit of a blow to the self-esteem of humanity. But what the point is, that was just the subroutines that are called into being. The program that's calling them into action is the rest, which had previously been written off as junk.
Well, this is another example of materialist building his case on facts that turned out to be wrong. Any reasonable person might wonder what else he might be 100% wrong about. Okay, I want us to spend a little more time considering what information is and where it comes from. While you're watching this video, keep in mind that there are 1.5 gigabytes of data in every cell of your body. Added up, you have 60 grams of DNA in your body. 1 gram of DNA equals 455 exabytes of data. How much is that? Well, 1 exabyte equals 100 billion gigabytes.
The crucial question that will decide the debate about biological origins is precisely the question of the origin of information. If you don't have instructions, if you don't have information, you can't build anything in life. Everyone who works in biology knows that information is needed in order for a living cell to do what it does. On the other hand, there's this huge mystery surrounding information, because we also know as humans that information doesn't come from nowhere, it has to come from somewhere. So you have this big question mark in the origins question in biology: where did all the information come from?
Until 530 million years ago, the oceans of the early Earth were almost completely void of animal life. Then, within a geologically brief span of perhaps 10 million years, the waters were suddenly alive, teeming with a riot of complex animals, representing most of the major animal body plans that have ever existed on our planet. Known today as the Cambrian explosion, this mysterious episode in life's history was familiar to Charles Darwin, who regarded it as a disturbing challenge to his theory of gradual and unguided evolution by natural selection. During the past century, the mystery of the Cambrian explosion has deepened as scientists have discovered the central role played by biological information in the history of life.
The Cambrian explosion is not just an explosion or the abrupt appearance of many new forms of animal life. It's also an explosion, or would have required a huge infusion or generation of new biological information. Biological form requires biological information.
Scientists' understanding of biological information advanced dramatically when Cambridge University researchers James Watson and Francis Crick made a startling discovery. They found that the structure of the DNA molecule stores information in the form of a 4-character digital code. Strings of precisely sequenced chemicals called nucleotide bases supply the assembly instructions, the information, for building the crucial protein molecules that living cells need to survive. Crick later came to realize that the chemical constituents in DNA function like letters in a written language, or digital symbols in a section of computer code. Just as English letters convey a particular message depending on their arrangement, the sequences of chemical bases along the spine of the DNA molecule convey precise instructions for building proteins. The arrangement of these bases directs the arrangement of the 20 different kinds of amino acids that make up protein molecules. Proteins in turn perform a vast array of critical jobs inside cells, catalyzing reactions, processing genetic information, and forming the structural parts of molecular machines and other biological structures. Building new animals requires many new protein molecules, and building new proteins requires new biological information.
I used to ask my students a question when I was teaching. If you want to give your computer a new function, what do you have to give it? And they would know. They would say code or software or instructions, a program. All those are the correct answer. To generate a new function in a computer, you have to have code. You have to have instructions. The same thing turns out to be true in biology. This is the great discovery of the second half of 20th century biology, that information is running the show in biological systems. To build a new form of animal life requires cell types, requires proteins, and therefore requires genetic information. And that's the big question that the Cambrian explosion presents. If you want to think about how to build an animal, how would these animals get built, you have to have some explanation for the informational requirements of their construction.
According to modern evolutionary theory, new proteins and new forms of animal life arise through random genetic mutations, sifted by natural selection. But in an alphabetic text or a section of computer code, random changes typically degrade meaning or functionality and ultimately generate gibberish.
As we've come to appreciate the digital or typographic character of genetic information, it raises some really interesting questions about the efficacy of that mutation-driven mechanism. And we know from computer code, for example, that if you start making random changes to a section of computer code, you're much more likely to degrade the information that's there already than you are to come up with a new operating system or program.
This problem has long been recognized by computer scientists, mathematicians, and engineers, including a group from MIT who convened a now famous conference at the Wistar Institute in Philadelphia in 1966. These scientists met to consider whether the random mutation natural selection mechanism could conceivably generate enough biological information to build a new animal or even a new protein in the time available to the evolutionary process. One of these scientists was MIT engineering professor Murray Eden.
No currently existing formal language can tolerate random changes in the symbol sequences which express its sentences. Meaning is almost invariably destroyed. Murray Eden, MIT.
Eden knew that random changes to alphabetic or digital characters inevitably degrade the information in any section of alphabetic text or digital code, and for a very good reason.
Whether you're talking about digital code in a software program or a section of text in an English sentence or book, or the genetic text in DNA, there are vastly more ways to arrange the relevant characters that convey the information in a way that will produce gibberish than there are that will produce function or meaning.
Eden and his colleagues suspected that the genetic code faced a similar difficulty. When it came to producing new genetic information, at least enough to generate a new protein, the random mutation natural selection mechanism had to deal with what mathematicians call a combinatorial problem. In mathematics, the term combinatorial refers to the number of possible ways that a set of objects can be arranged or combined. In genetics, the combinatorial problem poses a severe challenge to the random mutation natural selection mechanism. To understand why, imagine a thief who would like to steal a beautiful new bike. All that stands between the thief and the bike is a lock with 4 dials, each marked with the numbers 0 to 9. But there is only one correct combination that will set the bike free.
The reason a bike lock works is that there are vastly more ways of arranging those numeric characters that will keep the lock closed than there are that will open the lock.
A thief without knowledge of the combination must guess the right combination from among 10 times 10 times 10 times 10 possibilities. That's 10,000 possible combinations, which usually would be more than enough to defeat a random search for the one right combination. Yet there is still a way the thief might succeed. If he has enough time to try enough combinations, he might eventually identify the right one by chance. For example, if trying each combination takes 10 seconds, then in 15 hours an especially diligent thief could try more than 5,000 combinations, or more than half of the total possible combinations. In that case, he would be more likely to succeed than to fail in opening the lock. But now imagine a much more complicated lock. Instead of 4 dials, this lock has 10 dials. Instead of 10,000 possible combinations, this lock has 10 to the 10th power, or 10 billion possible combinations. With only one combination that will open the lock out of 10 billion, it's much more likely that the thief will fail, even if he devotes his entire life to the task. So what about relying on random mutations to search for a new DNA sequence capable of directing the construction of a new functional protein? Would such a random search for new genetic information be more likely to succeed or fail in the time available to the evolutionary process? In other words, is a random mutational search for a new gene or protein more like the search for the combination on the 4-dial lock or the 10-dial lock? The scientists at the Wistar conference were unable to definitively answer that question because at the time, no one could adequately quantify how hard the search problem was.
So in the late 1960s, one could make these arguments based upon analogy to things we understood, written language, computer code, but we didn't have any experimental data to show whether those analogies actually suited the biological case. So no one could come up with the exact numbers to answer these questions back then.
Molecular biologists at the time did know that the number of possible combinations corresponding to any given sequence of DNA is extremely large and grows exponentially with the length of the molecule in question. For example, corresponding to one short protein 150 amino acids long, There are 10 to the 195th power other amino acid arrangements of that length. That's an unimaginably large number. But scientists in the 1960s didn't know how many of those arrangements were actually functional. They didn't know, in effect, how many combinations would open the lock. That didn't stop evolutionary biologists from speculating. Many argued that there must be a high proportion of functional sequences among all possible sequences, so that a random search for a new functional sequence would have a high probability of success.
The way they did that is to say, well, maybe biological sequences are not nearly as finicky, not nearly as picky about which characters are where as written language is or as computer code is. And so that's the track that they took. That maybe proteins don't really care which amino acid is where, and there's a great deal of variability, and therefore you can have the same function performed by a huge number of protein chains and a huge number of genes.
But recent experiments in molecular biology and protein science have replaced speculation with data. These experiments have established that DNA-based sequences capable of making functional proteins are in fact extremely rare among the vast number of possible sequences. Just how rare? After working at Cambridge University, molecular biologist Douglas Axe set out to answer that question, using a technique called site-directed mutagenesis. His experiments enabled him to estimate that for every DNA sequence that generates a functional protein of just 150 amino acids in length, there are 10 to the 77th amino acid arrangements that will not fold into a stable three-dimensional protein structure capable of performing that biological function. One correct sequence for every 10 to the 77th power incorrect sequences. That's equivalent to searching for just one combination on a lock with 10 digits on each of 77 dials. To put this in perspective, keep in mind that there are only 10 to the 65th atoms in the entire Milky Way galaxy. Could random genetic mutations effectively search a space of possibilities that large in order to find even a single functional protein sequence?
So given that you have this very daunting probability, 1 in 10 to the 77th power, how could something that improbable happen? Well, as we know generally with improbable things, the way that they can happen is by having lots and lots of opportunities for them to happen. So for life, those opportunities take the form of individual living organisms in which a mutation could occur that could conceivably provide the solution. No matter how rare it is, if you get enough of these opportunities, it can become probable. So the question is, if the number is 1 in 10 to the 77th power, if that's the improbability that must be overcome, would the number of organisms that have existed on the planet since the beginning of life come close to matching that number? And it turns out that it doesn't come anywhere close.
During the entire 3.5 billion year history of life on Earth, it is estimated that only 10 to the 40th individual organisms have ever lived. Yet 10 to the 40th power represents only a small fraction of 10 to the 77th power, only 1 10 trillion trillionth to be exact. In other words, for even a single functioning protein fold to arise, the mutation selection mechanism would have time to search just a tiny fraction of the total number of relevant sequences, 1 10 trillion trillionth of the total possibilities. It follows that it is overwhelmingly likely that a random mutational search would have failed to produce even one new functional protein fold in the entire history of life on Earth. Of course, the building of new animals would actually require the creation of many new proteins. For this and other reasons, a number of scientists are now questioning the creative power of the random mutation natural selection mechanism. Even evolutionary biologists writing in professional peer-reviewed biology journals are acknowledging difficulties with traditional evolutionary theory. Some are willing to admit we already live in a post-Darwinian era, while others are calling for new theories of evolution. Meyer and Axe are part of a growing minority that has urged the consideration of another possibility. For Maya, the recognition of that other possibility grew out of his work as a PhD student in the philosophy of science at Cambridge University. During his studies, Maya ended up examining the scientific method used by Charles Darwin in his classic work On the Origin of Species. Darwin's method focused on trying to establish the causes of events in the remote past, in history. Darwin's historical scientific method is different from what many people ordinarily think of science.
It's a much more forensic style of reasoning than your ordinary experimental bench science. You're reasoning from the clues that are left behind, from the evidence that we have before us, back to the probable or possible causes that might explain what produced life in the first place, or what produced animal life, or what produced those clues that lie before us. And I ended up doing my dissertation in Cambridge on the historical scientific method. And one of the things I discovered in the process of my research was that there was this distinctive method. It has a name, and that name is the method of multiple competing hypotheses, or the method of inferring to the best explanation.
But how do scientists studying biological history determine which explanation is actually best? Maya found an answer again in the work of Darwin and his contemporary, the great geologist Charles Lyell, who argued that in explaining the past, the present is the key. Lyell insisted that we should seek explanations based on our knowledge of presently acting causes, or causes now in operation. All this led Maya to ask a critical question.
What is the cause now in operation for the production of digital information? Because the crucial question in the origin of animal life and the origin of life itself is, where did the information come from? Information stored digitally in the DNA molecule. Where did that information come from that is necessary to build these new forms of life? And I realized that the answer to that question is intelligence. The cause now in operation, the cause of which we know from our uniform and repeated experience, another key idea from Lyell, that is capable of producing information, is intelligence. Whether we're looking at a hieroglyphic inscription, or a paragraph in a book, or a section of computer code, or even information embedded in a radio signal, whenever we see information, especially when we find information in a digital or typographic form, and we trace it back to its ultimate source, we always come to a mind, not a material process. So the discovery that information is running the show in life, the discovery that there are these huge infusions of information in the history of life, such as the one that occurs in the Cambrian explosion, suggests that a designing intelligence has played a role in the history of life. And it also suggested to me that it was possible to formulate a scientific case for intelligent design, that is, a case for intelligent design based on the same scientific method of reasoning that Charles Darwin had used in The Origin of Species. And so if you want to say intelligent design isn't science, you would have to say that the Darwinian argument in The Origin of Species is also not science, but no one really wants to say that. He's not using an unscientific method, he's just using a different method of scientific reasoning, an historical method of scientific reasoning, and I used that exact same method in formulating the positive case for intelligent design in both Darwin's Doubt and in Signature in the Cell.
Bill Gates, one of the great minds in computer programming, has said this: DNA is like a computer program, but far, far more advanced than any software ever created. What do you associate with software programs today? Intelligence. What would happen to a software program if you made random changes to the code? Could you start with Pac-Man and make a bunch of random changes and then wind up with Call of Duty? Does it really make sense to believe that making random changes to the DNA of a bacteria would result in a human? As I mentioned in a previous letter, scientists have studied the quantity of harmful mutations in relation to beneficial ones and found that the bad ones grossly outweigh the good ones. Well, in case you'd like to do a little more study on this point, I'll include some other websites in the comments section. With that, I'll close out this letter. We've now looked at all of the main disciplines of science. So in the next letter, we'll take a quick review and summary of everything we've learned so far. Well, remember, I love you, sweetheart. I'm happy to hear your cold is better. Don't forget to call your mom, and I'll talk to you soon.